Can we record it, Peter? Peter, can we record it?
Yeah, sure. No problem.
Okay. So thank you very much, Peter, for joining us. We're honored to have you with us today. So very briefly, all about Professor Peter Karayiannis. He is a professor of microbiology, molecular biology at the University of Nicosia Medical School in Cyprus. He is former Associate Dean for Faculty and Research and co-chair of the Department of Basic and Clinical Sciences at Cyprus. Yeah. Professor Karayiannis holds a BSc and PhD in medical microbiology from the University of Liverpool, and thereafter he's been working at University of Liverpool, Royal Free Hospital Medical School, London, and St. Mary's Hospital, Imperial College, London for like several decades. He's on the editorial board of several journals, and currently he is European editor of the Journal of Viral Hepatitis. So, as you know, his main interests are in viral hepatitis, different hepatitis viruses, predominantly hepatitis B and C. And his work involves the deciphering mechanisms that favor viral persistence and evasion of the immune response during chronic infection, the effect of HPV variants arising during the natural history of the infection on long-term disease outcome, and those arising as a result of human intervention through vaccination or antiviral treatment, as well as new approaches to treatment. In addition to that, Professor Karayiannis has co-authored 228 original papers in peer-reviewed journals and has authored several chapters in the books. Currently, Professor Peter Karayiannis is a member of the advisory committee to the Minister of Health in Cyprus on the Covid-19 pandemic, the hepatitis elimination program, and the antimicrobial resistance. Now, without further ado, I request Professor Karayiannis to start his presentation and share his valuable knowledge on COVID-19 with us. Thank you, Peter.
Thank you very much, Fuzia, for that introduction. And of course, thank you very much for the invitation to give you a sort of an overview of the COVID-19 pandemic and where we go from now and how we can be better prepared for potential new pandemics in the future. So let me just share my slides and then I shall start my presentation. So I'll move straight to this slide here. The way I've structured my talk, I'm going to give you some sort of general information about coronaviruses. Then I will talk a bit about the epidemiology of the pandemic in Cyprus, worldwide, the variants, what we did right, what we did wrong, and where we go from here. So, of course, when the virus broke on the scene in November 2019 in Wuhan in China, one of the first things that was done straight away was to put samples under the electron microscope. And of course, the abundance of virus particles in the samples allowed the identification through the characteristic appearance of the virus under the electron microscope, as shown here, with the spike protein on the outside forming this crown-like appearance from which the name coronaviridae is derived from. So we knew that we were dealing with a coronavirus, and therefore very quickly, and in fact this was in time record, that the genome of the virus was deciphered, it was cloned, sequenced. And
sorry, Peter, can you go to the presentation mode?
Oh, yes, thank you, Fuzia. I forgot about that. Yes, thank you. Let's go down. So yes, the genomic organization then through the sequences that were obtained was very characteristic of coronaviruses, and which have 4 structural proteins which form the envelope as shown here. We have the nucleocapsid protein, which surrounds the RNA genome of the virus, which of course is single-stranded, positive polarity, which means that it can be translated directly into protein because it behaves like a messenger RNA. And of course, the virus makes a number of non-structural proteins, the most important one being the RNA-dependent RNA polymerase, the 2 proteases, a helicase, and at least 16 other non-structural proteins, which are extremely important in the replication cycle of the virus. And early on, it was established that the receptor on the cells on which the virus attaches is the angiotensin converting enzyme 2. So the genome of the virus, as I said, shows the characteristic coronavirus organization. There are 2 very big open reading frames, OF1A, OF1B, which are translated to give 2 polyproteins. OF1 is one of the protags, and then through slippage of the ribosomes to OF1B, we get a much bigger polyprotein that stretches to the end of 1B. And these encode for early proteins, as I said, mainly replication proteins, which are important in the early stages. And then there is a step during which a copy of the genome of the virus is made, which is negative sense, which then is used as a template for the production of progeny RNA genomes, but also for the transcription of a number of other transcripts as shown here, which encode for the structural proteins, which it is necessary to be produced later on in the life cycle to produce new particles. And because these RNA, messenger RNAs are produced later, this is where some mistakes in the transcription can occur, particularly in the spike protein here, which has given rise to the various variants of the virus, which we now know that as a whole, they constitute immune escape variants. The genome of the virus is the largest as far as RNA viruses are concerned, 30,000 nucleotides long. Through phylogenetic tree analysis, we now know that at least the 8 coronaviruses that cause human disease belong to the alpha and beta genera of the coronaviruses. The first 4 ones, as you can see here, are the ones that cause the usual colds or gastroenteritis in humans. And the last 3 are the ones which are much more serious. The original SARS virus, which appeared in 2004 in the Far East. The MERS one, which appeared in the Middle East around 2012. And of course, the SARS-CoV-2, which appeared in 2019 in Wuhan. And we know that at least the last 3 viruses I mentioned originate from bats through phylogenetic tree analysis again. However, there are intermediate hosts which may transmit the virus to humans. The original SARS was through civets, which they were being used or rather sold in markets in China, South China. MERS is from the dromad camels in the Middle East. And for SARS, there's still a big question mark whether the intermediate hosts are anteaters, snakes, or something else, or whether it has escaped from the lab following isolation of the virus directly from bats. And over on this side, we have the human isolates, which some of the intermediate hosts here may be domestic animals like cattle, sheep, but also rodents. So the life cycle of the virus is quite simple. Everything is carried out in the cytoplasm, being an RNA virus. So the ACE2 is the receptor. The virus is taken in, uncoated. You have transcription, translation. The proteins, the structural proteins are inserted in the membrane of the endoplasmic reticulum as shown here, and then of course the mature nucleocapsids pad off into the lumen of the ER, and from there on they're taken to the surface of the cells where they are released. So when SARS-2 broke on the scene, what did we do wrong? And we allowed the virus to spread so quickly around the globe and generate conditions of pandemic. Firstly, the Chinese authorities did not reveal early enough that there was man-to-man transmission. This was hidden away from the international community. We underestimated the dynamic of transmission of the virus. These are not figure which we use to denote transmissibility, was by far much higher than 1. In fact, in certain situations, it was estimated to be up to 15. In other words, a single person could infect 15 other people, and we used to refer to these people as super spreaders. The WHO was extremely slow in declaring a pandemic. It took 2.5 months before they went up to the highest alert point. The governments around the globe were completely unprepared for this type of emergency. There was not enough intensive care beds and equipment. People were dying in the corridor because there were not enough respirators. There were vast shortage of personal protective equipment, particularly during the first wave of the pandemic. The private sector, the private hospitals, in other words, were not involved in the emergency until later. Because of this, some people saw the chance to make money quickly. There was black marketing and hoarding of drugs and materials, creating shortages. There was also variable success in implementing the rules on the masks, the safe distancing, and hand hygiene. A lot of people just did not apply these rules. And one of the biggest probably problems was the lack of community participation and intersectoral coordination. In other words, there was complete disarray at the beginning of the pandemic. What else went wrong? The media went for the sensationalist side of things, creating panic. There was a lot of spreading of fake news through social media, particularly through Twitter and Facebook. And this was more intense when the vaccines came online, when they were demonized by certain sectors. There was lack of government-directed explanation of the measures, so this created a lot of resistance in the community. And of course, what we all underestimated was the immense impact of the pandemic on the economies of various countries. The social structure, we all saw the very strict lockdowns in China and how people were locked in for weeks on end. And also the mental impact, particularly on the young, the children who couldn't get contact with their friends and school colleagues. What did we do to respond to this emergency? Of course, in an attempt initially to restrict transmission, we had to enforce these lockdowns, which were of variable duration depending on the country. We had to enforce the use of masks indoors during working hours and outdoors when out shopping or going about our business, keeping a safe distance of at least 2 meters and exercising hand hygiene. Governments acted by setting up approved hospitals or wards in hospitals entirely for COVID-19 cases. And this, of course, had a negative impact on chronic diseases, operations, cancer patients, and so on. As new information was generated, guidelines were updated frequently. People require medical attention. They did this through telemedicine. Businesses adapted to doing that through online and home working. And of course, schooling, primary, secondary, and tertiary education was adapted to online teaching. Perhaps one of the best responses was the development of vaccines to combat the disease. And this became possible through the very quick sequencing of the genome, which allowed in the first instance the formulation of diagnostic tests through PCR and later on through the rapid tests. This information was shared with scientists around the globe very quickly, and these allowed the development of candidate vaccines, whether mRNA or virus vector-based vaccine, and later on protein-based vaccines, to be formulated, tested in parallel to save time, which allowed the vaccine to come on the market within about a year of the appearance of the disease. Vaccination strategies varied depending from country to country. There was inequality, however, in vaccine distribution. Richer countries got the vaccine first, poorer countries much later. And then, of course, there were a lot of obstacles in the vaccination programs. Demand was high. You got several dosages out of the same ampule. This sometimes led to wastage. Access was limited in certain countries. Supplies were slow. And of course, hesitancy. A lot of people were afraid to be immunized with something that they didn't know much about. And the other thing about the assessment of the efficacy of these vaccines was that they were given. One immediately assumed people were immune because there was no antibody testing to confirm that. Let me now show you what happened in Cyprus. Cyprus is a very small country, only about 1 million people. You can see the first wave is hardly visible with this sort of star here. However, subsequent waves, as you can see with the different variants, beta, alpha, delta, and then the Omicron variants were much more severe. The most severe was the Delta one, and as I will show you in a minute, that caused the majority of deaths. But massive transmission during the Omicron waves, which, however, as we now know, the virus, thankfully, was less pathogenic. Most of the population was immunized, so the impact of these waves was not as great. This slide here shows you the succession of the Omicron variants in Cyprus over time, starting with the Delta here in black, which was the most serious, giving way to the various Omicron variants. The death rate went up over time, but as you can see here, up to now, we only had 1,330 deaths and nearly 650,000. In other words, 2/3 of the population contracted the disease eventually. But I think as a country, we did extremely well in managing the pandemic and restricting the death rate. And this here shows you when vaccination in Cyprus started early in 2021, in January, actually. And by 15 months later, we had 69% coverage rate in Cyprus as far as the vaccine. And from then on, the death rate went right down because the hybrid immunity, in other words, infection and vaccination, did quite well in protecting people. I accessed these 2 days ago just to show you what has happened in the world. I urge you to not rely too much on these figures, because now with the rapid testing, a lot of the cases are not been notified. So this is from the Johns Hopkins database. Nearly 700 million people have been infected and nearly 7 million have died. But if you look at the times estimate, this is put at around 20 million. So this is a gross underestimate of the people who have died as a result of COVID. And of course, the plus on this is that there have been 13 billion immunizations so far. And this is shown better in this slide. I've only put it here to show you that the death rate has been going down and the vaccination rate has been going down. Hopefully, these will not cause trouble, because people are more reluctant to get vaccinated when they don't hear there are large numbers of infections, and we don't know what impact this is likely to have on new variants in the future. So when we talk about variants, just a couple of words. These are defined through sequencing, and they are categorized depending on whether they are just to be monitored, whether they are of interest, or whether they are of concern. And what do we mean by that? We mean variants that have increased transmissibility, as it has happened with the Omicron variants. If they have increased disease severity, thankfully, this has not happened with the Omicron variants. They are less severe variants. And whether we have an impact on the diagnostics, in other words, whether the treatments we have, and particularly the vaccines, are no longer effective because the mutations they throw up are in the surface proteins in regions which are recognized by neutralizing antibodies, and therefore making the vaccines less effective. Where did these various variants originate? At least the Beta and Omicron variants, they originated in South Africa and then they spread around the world. The Alpha variant in the United Kingdom, and the most dangerous one, the Delta one, in India. What has happened to these variants? They have all disappeared from the face of the earth except from the Omicron. And because of this, I am a bit relieved that if any new variants come up, they will be on this genetic background. So I hope they are unlikely to be more pathogenic than anything we've seen so far. I put this here just to confirm what I've just said. The original Omicron variant is here, giving rise to all these new clades of the various types of Omicron which we've had, including the more recent ones like Kraken, the Arcturus, the Centaur, and so on. What is the likelihood of something similar occurring in the future? What reasons might there be for this? Reduced immunization coverage. And we're beginning to see this happening with very good vaccines like the polio and measles vaccines. A lot of parents don't immunize their children. Occasionally, we have outbreaks with these diseases, causing fatal cases. Easy population movements. There's a lot of refugees seeking livelihoods elsewhere. The climatic change, we're seeing so many changes in recent months with heavy rains, very hot weather, and so on. What precautions must we take? We should maintain the immunization coverage, have effective surveillance, be on the lookout for anything new, and maintain the safety measures. And what are the likely pathogens? In the last few months, we were alarmed to see monkeypox get out of Africa and move into Western countries, a disease that thankfully did not cause many deaths. Cholera has shown its face in many countries because of bad hygiene, either earthquakes, floods, war, forcing people to live rough under very unhygienic conditions. Then we've got a lot of viruses, particularly the hemorrhagic fever viruses, which are transmitted by vectors like mosquitoes, ticks, etc., which, because of the change of the climatic conditions, we now see, for example, West Nile virus making inroads into Europe. I'll show the slide in a minute, but this slide here I put to show you that over a few months with the monkeypox, you can see how the virus has changed as it spread in humans and how possibly it began to adapt in humans, and how it was trafficked around the globe. Air travel these days can allow somebody who's infected to spread the virus from one end of the world to the other. I mentioned just before West Nile virus, which was restricted to Northern Africa. You can see now that it's making inroads into Southern Europe, Turkey, because the mosquitoes that carry it are now endemic in these regions. In the United States is a similar problem. Normally the virus is found in birds and the vector is the mosquito, as I said, which can transmit the virus to horses and to humans, which are dead ends. There is no man-to-man transmission. So these sort of viruses are unlikely to cause a pandemic because there's no man-to-man transmission. However, these will require restriction of the mosquito populations if we are to prevent a spread of these viruses. It kills the birds, it infects, as shown here. And then again, more recently, we've hearing with increasing frequency outbreaks of the most dangerous and deadly viruses which belong to the filoviridae, such as Ebola and Marburg. And the death rate here is 25 to 90%. And we don't have any antivirals. There are some potential vaccines being trialed out, but if these viruses get out of Africa, you can see how extensive a problem it might be. There's another group of viruses, the Flaviviridae. Hepatitis C belongs to these viruses, but the majority of other viruses are transmitted by ticks and mosquitoes. And a lot of these Flaviviridae are found in bats. It seems that the bats host a whole array of different viruses. And we as humans like to explore caves where bats tend to sleep at night. So the potential for diseases to come out of these caves and be spread to communities is real. However, where I think the next danger lies, and possibly the next pandemic, is the bird flu viruses. Up to now, transmission to man has not been easy because we don't have the appropriate receptor. However, as shown here, recently and in the past, there have been transmissions to humans. In Cambodia, for example, very recently, there were a handful of cases. And they've noticed that these viruses, they caused mutations in their genome to allow them to replicate better in eukaryotic cells. So the danger lies in these viruses adapting to humans and then been transmitted quite easily as the coronavirus through the respiratory route, from man to man also. So we need to be on the lookout here. And just to conclude, what we need to do, we mustn't let our guard down. We must not forget the lessons we've learned from the COVID-19 pandemic. The national health systems around the world must be prepared. The government must put the money in maintaining the intensive care units, having enough respirators, enough personal protective equipment. Scientists themselves must keep an eye on anything that's circulating through sequencing, keep an eye on the viruses and how they change. And then the pharmaceutical and vaccine producing companies should be on the lookout for new approaches to develop efficient treatments, and that applies to vaccines as well, as quickly as possible. All that remains is for me to thank you for your attention, and I'll be happy to answer any questions you may have.
Thank you very much, Peter, for a wonderful presentation. Some questions, you did mention about climate change and, threat of other viruses. Like, for instance, okay, so France is dealing with a dengue virus because we never had mosquitoes there, but now they do. But in Pakistan, India, Bangladesh, this part of the world, we always have dengue virus, it's been ongoing for decades, and chikungunya as well. So you can't differentiate between the symptoms, the joint aches or whatever. So, but and as you rightly said, when people fly, they can carry the infection there. So don't you think that if that is the case, the climate change is there, we have that problem in Pakistan as well, but people have been flying out, flying in, and I don't understand why couldn't we transmit more, mosquito eggs or something to Europe earlier, like it's only happening now in Europe.
Let me tell you what happened in Cyprus this past winter. For the first time, we detected the Aedes aegypti mosquitoes in Cyprus, which theoretically can carry a lot of, how they came, we don't know, but they were detected, and immediately what happened here, they released sterile male mosquitoes in an attempt to restrict the reproduction, spraying, and so on. But at the moment, we don't have in Cyprus any carriers of the diseases like dengue, yellow fever, etc. So these mosquitoes are unlikely to become infected unless a carrier from another country comes to Cyprus with infection, and then we will have a problem. And of course, the climatic change is allowing these mosquitoes, which were not indigenous for many years, in countries of not just Cyprus, but Southern Europe as well, now to have the potential to become infected and then cause spread of whatever virus comes to Europe. This has happened with West Nile virus. So it may very well happen, not just with viruses, but also with malaria, with parasites.
Yeah, that's right. Yeah, thank you, Peter.
The other thing I wanted to make, sorry, Fusia, another important factor is that you mentioned that you have these diseases there. So you're an endemic area, your people have got some sort of immunity in the population. In the Western world, there's no immunity whatsoever. The ground is virgin for the spread and for the manifestation of most severe outcomes.
That's a valid point. But then at the same time, with dengue virus, like different serotypes. So because it's hemorrhagic fever, so, we can still have deaths. But, that also, the good point is that, in the Europe, they don't have immunity against these viruses. So yes, they are kind of, they're immune because they haven't been exposed to this, so they don't have the immunity. So if worse come worse, if we have an infection in the Europe and that part of the world where they haven't seen this dengue virus or chikungunya, they are more likely to die than
Yes.
this part of the world, our
Yeah.
part of the world. Oh, it's very tricky. Peter, you did mention, different variants of coronavirus, until this Omicron popped up. And you mentioned that, the other variants, Alpha, Beta, Delta, they disappeared from the world. What could be the reason for them to disappear?
It's, I think, as immunity was building up, they had to change. And in order to do that, they had to either opt for the easier transmissibility, which they've done, and reduce pathogenicity. A pathogen that kills its host is not a successful pathogen. Okay, the pathogen wants to perpetuate itself. So it wants its host to remain alive. And of course, the plus thing for the virus here is that natural immunity and vaccine immunity wanes over time, allowing the virus to infect and reinfect. And we know this from not just the different coronaviruses, but also the different rhinoviruses. We have common codes because these viruses change into different types, etc. And therefore, they have the chance to persevere and remain with us as long as man is around. So I feel that eventually the coronavirus will become a normal cold as the other coronaviruses until something else comes on the scene.
That's very interesting. Like viruses are much more cleverer than what we think of them. They're
Yes,
tiny little bits and causing us so much of a problem. So Peter, I had a question as well. Like when you, she has a question. Sorry, we have a question probably on WhatsApp. So there's a question from one of the attendees. And the question is, what precautions should we take to save our poultry from bird flu? Right.
Well, that's a very good question. Poultry, there are vaccines that can be used. And there are vaccines, for example, to protect poultry against Newcastle disease virus. The same thing can be done for farms, etc. But how can you control infection in the wild? How can you control birds which migrate from going from one country to the other? You can't. It's just an impossible task. That's why when there is an outbreak, usually they opt for the mass killing of the birds from the farms in an attempt to restrict transmission to other farms. But as I said, usually it's geese, ducks, etc., which also fly from country to country that can transmit the virus. And there's nothing you can do there.
So we just need to be prepared for
and vigilant,
and have drugs for ourselves, for humans.
Yes,
that's much more.
Yes. At least with the flu viruses, we've got the oseltamivir and Tamiflu, which are effective. They are not used in combination. They're monotherapies. There have been resistance against these two drugs. That's why they're not used widely. But the other good thing there is that we have a huge experience with flu vaccines. We can produce a vaccine very quickly.
Yeah. Is there a question? There is a question here. somebody would like to ask a question. Can we allow it? Okay. Message. Okay. Sorry, the question is there any oral pharmaceutical company in pharmaceutical drug in clinical trials for COVID treatment, like the oral, like any tablet or anything?
Well, other than the initially we had the remdesivir, then we have the molnupiravir, and now we have the I've forgotten the name of the last one, the most effective one, which is more direct. I haven't heard of anything else being developed at the moment as far as COVID-19 is concerned.
Okay, we have a raised hand, so can we allow the person to ask the question directly? Could you unmute yourself and ask the question, please? Nawal?
Hello, can you hear me?
Yes, speak a bit
Yes,
loud, please.
Can you hear me now?
Yes.
Yes, go on, please.
So my question was, what were some key lessons learned from COVID-19 pandemic, particularly regarding public health preparedness and response?
Fuzia, there was a bit of,
Yeah.
did you have the question? Can you repeat, please?
So she said, what lessons we learned from COVID-19?
Response and
Please read it from the chat box.
Okay.
Particularly regarding...
Like our preparedness for the next pandemic?
Well, if this has got to do with the lessons we've learned, I think I've mentioned this in my presentation, and I also stress that we mustn't forget this. And I think the WHO recently issued a warning to governments not to undo everything that they've set up in an attempt to fight COVID-19, because the next major issue may be around the corner. Obviously, we all hope that this will not be the case, but at least we mustn't be caught again unawares.
Yeah, so the question was, what were the key lessons learned from the COVID-19 pandemic, particularly regarding public health preparedness and response?
Yeah, well, I mean, we didn't have enough nurses, for example, trained for intensive care to cope with the problem. We didn't have enough respirators. We didn't have enough gloves. People, I mean, here in Cyprus, they used to put on dustbin liners to protect them when we didn't have nylon aprons, for example. And the same thing for overshoes, you know, nylon bags, which you use for shopping.
So Peter, you know, like this pandemic came after a century. And because people would have died and we were not prepared for this one. So do you think the next pandemic is after another 100 years, or it's just around the corner? You know, we know how to deal with that, but, you know, maybe 10 generations down the line wouldn't be prepared.
I mean, we've had other pandemics since the 1918 pandemic you're referring to, the Spanish flu. That was the biggest killer. It killed 50 million people. But there followed at least 3-4 other pandemics with flu viruses. But by the time they appeared, we had already created vaccines. There was immunity in the population, at least, which made the disease milder if you were re-exposed. I remember catching flu when I first went to the United Kingdom. And I never felt so sick in my life, you know. My whole body was aching. But touch wood, since then, I've never caught flu again. Or at least if I caught it, I passed it not so badly, you know. But with the H5N1, we don't have any immunity. We don't have any immunity against bird flu at the moment. So if it changes and it transmits to man more easily, it may be a different question then. But as I said, we have the technology, we have the know-how to act quickly. It's just amazing that we rolled out a vaccine within a year.
Yeah, they did a great job. It was like we were not very hopeful, but it just worked. It was a miracle to have vaccine available. There is somebody's raised a hand, so maybe that person can ask a question, and then I have another question in my WhatsApp. So Abbas, please could you unmute yourself and ask the question?
Yeah, sure thing. Hello, how are you all doing?
Hello.
Good, thank you. Go on.
So I wanted to ask about the climatic effects of the COVID. So we do not understand very well that how it performed in different climates, but I would like to know, based on the epidemiological data, that how it affected in different climates, in the hot weather and in the cold weather. And based on that percentage, how can we predict that the Omicron variant will have a boost in certain environments and in certain climates as well?
Okay, very good question. I must say that the first wave of the first year of the pandemic, shall I say, we all expressed the hope that this coronavirus will behave like all other respiratory disease viruses. In other words, be seasonal, appear in the winter or say in the autumn-winter time, and then disappear in the summer. This doesn't seem to have been the case with this particular virus. We still have infections even during the summer. The reason that they are not in high numbers is because of the hot weather. We tend to be outside. We are not in enclosed areas so that the virus can linger within and us, breathe in contaminated air. And therefore, we've seen with this virus an increase in infections as we sort of become enclosed in the cold months indoors. We don't know how it's going to behave this winter. I suspect we will continue as we are. We will be seeing sporadic cases, but nothing like the waves we've had in the past. How it will behave in the future, perhaps eventually it will become seasonal.
Thank you, Peter. And another question. So they've asked, does COVID affect someone more badly if they have sinusitis?
We know that COVID is worse in people who have respiratory problems, whether it's asthma or cardiopulmonary problems. Smokers initially we thought would be worse off, but that didn't pan out from a number of studies. We don't know why some people were asymptomatic whilst others they succumbed very quickly. There are studies now indicating that there are polymorphisms in genes like FOXP3, for example, which may affect the intensity of the immune response, because we think that people who die, they have this cytokine storm effect and multi-organ failure. So if you respond drastically against the virus, you maybe damage your vital organs. Things that we knew, for example, me as interested in hepatitis B and C, we knew that certain genetic polymorphisms were determining whether you became chronic, whether you responded well to antiviral treatment or not, etc. These things seem to be translating to other infections now. So it's just a matter of time before we know which genes predispose you to a worse outcome.
Thank you very much. And I think that's all. These are your questions. Thank you very much, Peter, for your time. And thank you to Dr. Kastabani who co-moderate. He's been passing on the questions to me. It was a wonderful insight session. Thank you, Peter.
Okay. Fouzia, thank you very much for the invitation. It was a very stimulating conversation we've had, and the questions very much to the point. So I hope that we've all learned something out of not just the pandemic, but also out of this presentation.
Thank you very much.
Have a good day. Bye-bye. Bye-bye.
Bye. Take care. Bye.